Hello, welcome to Carousel. I'm your host, Barry Kesselman, and I hop on the carousel horses and bring you interesting people on interesting subjects or interesting projects. And we were so fascinated last time we met with Dr. Marty Yellin that we're going to continue our conversation with him and go on to some of the activities that he's been involved in since his initial activity. He was involved in a project called Hexagon, which was a spy satellite back in started in the 1960s. It went from 71 to 86, was just declassified in 2011. And it's the satellite that took film pictures of the Soviet Union and China for 20 missions. And if you are really interested in it and it's fascinating, go back and see our previous show. But I do want to ask a few more follow-up questions of you, Marty, so let me introduce you. Dr. Marty Yellin.
Thank you again.
My pleasure. We talked about the project and we'll delve into it, but today we're going to focus in a moment more about things that you're doing since that project.
Right.
Now, when you worked on that project, you worked for PerkinElmer. It was a CIA project, but you weren't part of the CIA.
No.
However, you operated within their aegis. And even though you were a super tech spy, I assume that at one time or another you became like a human spy that actually traveled in spy-like manners. Is that right?
Some of the PerkinElmer people were stationed overseas, and invariably once or twice I had to go overseas to gather information. In other words, in order to know what we were building, we had to understand what we were looking at. And that data was classified. We had some special facilities in countries that could look at Russia that were camouflaged as observatories looking at the stars, like for astronomers. They were really watching missile launches from Russia and other things and collecting signature data on reflectivity, color of the plume, and other things. I mean, they had this down to such a good science, they could look at the smokestack and know what's being made inside the building.
Wow.
By the color of the smoke coming out, which they could do optically in spectroscopy. So yeah, I had to do some of those trips. Of course, when I traveled, I couldn't say where I was from. I had my own name. I had one passport. It wasn't like, the Bourne identity where every time he goes to an airport, he opens a locker and takes out a new passport.
Oh, that's really disappointing to hear, but I understand.
Yeah, but it was a little nerve-wracking. I always had a military group with me. One sad thing that made the cover of the New York Times, we had a guy my age working the job who was offered a job in Iran to help run one of these. His name was Carousel, and they gave him a big financial incentive that they pay for all his college kids. He had 3 or 4 kids. He went there, he was there 2 weeks, and while driving to work, a Volkswagen got in front of him and one behind him, and they machine gunned him to death.
Wow.
Somebody must have figured out what was going on. Russia had him killed.
Wow.
And this is when the Shah was still in control. This is not when the Ayatollah took over. Then it got really bad. A lot of our people were killed that were over there gathering data when the Shah was overthrown.
Well, all the more reason to have Project Hexagon, which is a satellite, 60 tons?
It weighed 60,000 pounds.
60,000 pounds, about 80 or 90 miles above the Earth,
right.
Taking film photographs on 6x6 pieces of film,
yeah.
Focused down strategically to the Soviet Union and maybe China or wherever, and having at 200 frames per second, having a resolution
200 inches per second.
Each
frame was 6 inches.
Yes.
And it moved 200 inches per
200
second.
inches per second.
Yeah, so it's like.
And each frame could take a picture of about how many square miles or miles?
Well, one frame was 370 miles, like from here to Cincinnati, and it could see to 2-foot resolution. In fact, when the program was over, the last satellite flew over PerkinElmer in Danbury, and we could see our cars with our license plates. Couldn't read your license plate, but you could tell it was your car. You could see who was at work that day and who wasn't. That was amazing. That was from 90 miles up.
That is amazing.
Amazing, yeah.
And then the project eventually ended. That was
1986, digital photography started to come in. It wasn't CCDs yet, it was individual photodiodes. And of course that had the big advantage, you didn't have to wait. You didn't have to send anything down. You could permanently station these in space, because eventually it would have to come down. You couldn't put them that far away that they would always stay up there. They would have to be like 22,000 miles away, and you'd never get to see anything. So they were still at about 90 miles. But they were digital. So now you could sit in one room in the Pentagon and point to the Suez Canal, or point to Syria, or point to anywhere, Iran, and see what's going on.
So for the people who didn't get to view the first show, just tell us again how we got this film from 80 or 90 miles above the Earth down to the people in Washington who had to analyze it.
All right, there was 60 miles of film in each of 2 cameras. That's 120 miles total. And each camera had 4 take-up reels, so each held 1/4 of that 60 miles.
Right, and how did they get it down to Earth and capture?
So every time you filled up the first bucket, we called it, the first reel, the film would get cut and spliced to the previous reel, and then that bucket was in a canister with a rocket on it and a parachute, and it would shoot down from the bottom of the spacecraft to Earth from 90 miles out, which is pretty far. It's like 540,000 feet. 2 planes flying at 20,000 feet with a cable between them had a hook. There was a hook on this parachute, and they would try to catch it, which they missed the first time, but they never missed again after that for 35 times. And then they would reel it in and bring it to a special facility where it was developed, processed, and sent down to an intelligence group in Washington for analysis.
Amazing. So the optics you developed for the government must have been really helpful after the program finished.
Yes, well, the optics, and remember, we had to simulate space. We didn't know the effects of a launch, which is a lot of vibration and shock and G-loads, thermal vacuum, the temperature is minus 230 degrees. We had to simulate all that with giant chambers on Earth that would take a 60-foot long vehicle and test it. Having that, it allowed us to then be, excuse me, I love to cross my legs. We won the Hubble Space Telescope as a follow-on, which was not classified as strictly scientific. We had all the facilities for testing it and building it there. We built the optics and the pointing system, and that's still up there after 20 years. It was designed for 10 years, but that was sent up by the shuttle. Mm-hmm. It's not 90 miles up, it's lower. So the shuttle could bring it back into the shuttle, change things, change instruments as technology got better over 20 years, change the optics, change the instruments, replace the gyros, new solar panels, etc., and then leave it out there again. And it was designed to be maintained in space, whereas these couldn't. I mean, we didn't have a shuttle.
But the technology that was developed by you and others was what was used as part of or basis for the
technology of that first program led to things like the GPS. It led to so many things, and it was all just hard engineering, but it was the first. So.
You talked in the.
Oh, by the way, I want to say that this program was declassified, as you mentioned, just 2 years ago, less than 2 years ago. 12 years ago, as we were dying off, the remaining people said, our kids will never know about this because we couldn't tell anybody. So we decided to write a book that the CIA gave permission to. They kept the book. They gave one of the guys a special computer where everything was encoded. We would send our stuff to that computer, and he would edit it. The book is going to be published next month, in a few days actually, and we all can't wait to get it because 12 people essentially wrote it, and we'll sign it and give it to our kids and grandchildren as a goodbye present.
So that's going to be very interesting, especially to the people who know your story. Yeah. What's the name of the book?
I think it's called Hexagon, We Met the Mission, or something like that.
And the publisher?
The publisher is AIAA. American Institute of Aeronautics and Aerospace, Airspace and Aeronautics. And
a particular author for anyone who's looking this up?
Well, the guy who put it together is named Phil Pressel, but it really is a compendium of 12 of us writing their piece of it, in mechanical, electrical.
I look forward to having one and bringing it to you to get an
If you want to go to sleep early one night, read it.
That's very interesting. You talked about spying and classification and declassification, how difficult it was and how strict it was. And so I wonder if you could give us a bit of your thoughts on the current situation we have in spying, namely Mr. Snowden.
Oh, Mr. Snowden.
Not so much spying, but releasing of classified information.
Now, he works for the facility under the National Reconnaissance Organization responsible for collecting ELINT, it's called, electronic intelligence. That would be email, telephone, cell phones. Of course, when he joined that company, he had to sign a clause like I did, saying that anything he learned or did was highly classified and it was a violation, and they told you the rules. I mean, it could even be death, depending on what it was. So no matter what his political persuasions, I understand his viewpoint that maybe we should not have spied on civilians, although I personally know that having done that has prevented over 60 terrorist attacks in this country,
wow,
which are not going to be published. So more importantly, by him going to Russia, I'm sure they could find out anything they want, not only about what he knows, but how we gather the information, what our technology capabilities are, so they could change their codes. And that will really set us back 20 or 30 years. It really is bad. I think what he did is a terrible thing, and I'm a pretty liberal person at this point in my life. But he should have quit and kept his mouth shut and stopped, not jeopardize the lives of many people, which he will if he keeps talking the way he is.
Very interesting, very scary. Now, I want to switch gears and talk about what happened to you or what you've done since you're finished with the this kind of technology, spy satellites, Hubble satellites, and that. You've kind of switched over. Your background is engineering, and you have a doctorate degree in
biomedical engineering.
Engineering.
Yeah. When I was going for my doctorate, I always wanted to be in medicine. My mother died of cancer when I was like 13. My sister had cancer. She didn't die. My wife had cancer. 2 wives had cancer. My daughter had cancer, fortunately survived. When I started working on that classified program was when Richard Nixon had started the war on cancer in 1965. Unfortunately, the same amount of people that were dying of cancer then, about 300,000 a year in this country, are still dying of cancer. No progress was made. And you compare that to heart disease, where 80% deaths have been reduced in that time. Now, you could argue that cancer tends to be a disease of the older people. So people are living longer, so there's a larger population, and that's partially true. But it was a basic failure. The whole approach of chemotherapy, poisoning your body and hoping the cancer dies before you, radiation, burning you and hoping the cancer dies before you, and cutting it out, which is too late already if it's spread, that's not going to save your life, was barbaric. It's like leeches. We look back on leeches.
Yes.
It was only until they understood the genome, the Human Genome Project, and got into the genetic signaling of how the body communicates to its cells, that we began to understand what happens in cancer.
Now, my understanding, and I'm really a layman on this, but my understanding is cancer is not just cancer, not just a single thing. It's hundreds of different diseases, some genetic, some environmental.
Well, cancer, there's hundreds of genes involved in cancer, and you and I could have the same cancer, but it would be a different combination of problems. But all cancers have a known sequence of events, and that's why it takes a long time to develop. And it's almost like a slot machine with 6 wheels. And every time a lemon comes up, that's a bad mutation. And when you get 6 lemons, you're dead. I mean, typically it starts by the first line of defense. Now again, all this was learned in the last 20 or 30 years, but it's finally being put to fruition with the development of new drugs like Gleevec. And I'm not doing a commercial here. That really will take people with leukemia, that would have chronic myeloid leukemia, which would have been a death sentence 20 years ago. That now they could probably live their whole life like a diabetic as long as they take the drug. And new drugs are coming down because eventually the cancer gets smart, since it's always mutating, and it'll figure out how to overcome Gleevec, just like antibiotics with bacteria. And so the first line of defense is gene that's in everybody's cell. It's called P53. It's called a tumor suppressor gene, and it acts like the brakes on a car. Every time a cell reproduces, it checks to see if there's any mistakes. And if it can be fixed, it fixes it. But if it's really bad, it says commit suicide. It's called apostasis. It kills the cell so it won't spread. And P53 is great, because, and if it stopped it at that point, you'd never have to worry. Okay, unfortunately, some people are born, you get 1 P53 from each parent. Some people are born with 1 instead of 2, so you're more susceptible. Some people are born with none. They tend to die in childhood cancers.
Just how many genes are there in the
There's 26,000 genes when we broke the human genome, And what a gene is really is an instruction set to make a protein. You know, each gene is a code for an amino acid, and each protein consists, and I think there's like 16 or 20 different amino acids, and you just add them up and you build these three-dimensional models. That's what a protein is, and their shape determines what they do. So like on a cell, you'll have openings that certain proteins could fit into, and that's how they communicate. So
this
26,000 genes, but when we finally broke the human genome, we found that it looked like only about 10,000 seemed to do anything. In fact, the rest were called junk. And we assumed they were like viruses that hitchhiked a ride on us, because our viruses. Also, you realize that our body, which consists of 100 trillion cells, has 10 times as many non-human cells. We have 10 times as many bacteria and viruses in us than human cells. So a lot of their DNA is mixed with us too. And we couldn't live without them. It isn't that they just digest our food. They make vitamins, they make hormones, they make brain function. They really make us live, and they protect us from dangerous things.
And one of these 26,000 genes is the P?
Is the P53, which, and every cell has the same genes.
Right.
That's the other thing. I mean, you understand that when an egg gets fertilized, every cell is identical. And that's leading to what we're about to talk about, epigenetics. So when they looked at the genome, only 10,000 or so did anything. The rest they call junk. And in the last 6 years, they discovered it's not junk. They're switches for those genes that do something. Huh? They tell them when to turn on and off. So when you have an egg that's fertilized, all right, now one part of it, and all the cells are identical until about the first week. And now somebody's got to be the outside, somebody's got to be bone, heart. They all have the instruction set in every cell to make anything. It's like having the entire library in front of you, and it says, no, just go to volume 12, you're going to be a bone. Go to volume 3, you're going to be a heart. So these genes, these epigenes, switch the genes on and off to tell them to make things. Mm-hmm. But the most interesting thing, and they do most of their work when you're developing, but the most interesting thing is all during your life, these epigenes are doing things like that, which can make you get cancer, and it's controlled by your environment. How you eat, stress, exercise, all can switch these on.
Now, if you had a pair of twins.
Yeah, sorry, what?
If you had a pair of twins.
I was just about to say, you take two identical twins, identical. They have the same genome at birth, and they've done this experiment. In fact, they did this before they even broke the human genome, and they never understood why. At 60 years old, one has cancer, one doesn't. One has arthritis, one doesn't. One is a genius and one isn't. And what happened? I thought you're born with these genes, you know. The point is different genes came on and off that shouldn't and made them susceptible to certain illnesses. Now, most cancer is not inherited. It's very rare that you're born without the two P53. I'd say that we know that there's a couple of cancers caused by viruses, you know, cervical cancer, mm-hmm. and AIDS. And there's a couple of cancers, maybe one or two, caused by bacteria, pylobacteria, which causes the same thing that gives you ulcers can cause stomach cancer, but it's rare. And it's a bacteria, it's not stress that causes ulcers, they learned that.
Yes.
But the rest are the environment. Obviously cigarettes, asbestos, cleaning fluid, benzenes, all these chemicals that you eat and breathe in and drink that we don't know what they're doing have an effect.
Do we have any idea, take benzene or whatever else you want, how it works or how it affects us?
Yes, benzene is a classic example. I told you P53 is the gene that makes this protein that protects it. It has a certain shape. Mm-hmm. Benzene has a certain shape that blocks it. It locks onto it like a Tika toy and prevents it from using. You breathe in the fumes, it gets into your lung, it gets into your bloodstream, it goes to every cell in your body and blocks P53. Now you have a car without brakes. So that's the first wheel in that wheel I talked about.
Yes.
The second wheel is now, I have a stuck accelerator pedal. You're okay, so you don't have brakes, but the cell will only reproduce when it needs to, which is depending on what it is. I mean, skin cells, intestinal cells, they reproduce rapidly. That's why when you have chemotherapy, which kills rapidly growing cells, you lose your hair, you get indigestion, etc. And they'll come back. But most cells maybe reproduce once a year, unless you have you do damage. You cut your finger, stem cells, which are always in your body, will come back and make the skin and grow and close it up and then go back to bed.
Okay, so this benzene comes and it stops.
It stops P53,
so
but
there's
there
no
are
brakes.
other things that come in that turn on what's called oncogenes. When you're an embryo, you're growing rapidly. These are all oncogenes turned on to make the heart, the lungs, the bones. You don't want them on anymore, except if you do some damage and you got to rebuild your bone. Like if you break your ribs, sometimes the old ribs get eaten up by your body and it makes new ribs.
Wow.
Okay. Well, other drugs like cleaning fluid, which not only blocks P53, it also turns on some oncogenes, different ones in different cells. And now you have a car where the accelerator pedal is stuck.
That's 2.
So you have no brakes, and you're going like crazy. So now you're reproducing fast, okay? That's dangerous because you only get the mutation when you reproduce. As an example, throat cancer. You could smoke cigarettes, which is carcinogenic, but if you drink, the alcohol kills the cells, because alcohol will kill them in the lining. They'll start to grow rapidly to replace it just when the smoke is hitting them. See, when it's growing, it gets hit by the mutation, and that leads to throat cancer. So if you just drank and didn't smoke, I'm not saying that's good for you, or if you smoked and didn't drink, you'd have a better chance of not getting throat cancer. You might get bladder cancer, lung cancer, and everything else.
Okay, so I shouldn't smoke, I shouldn't drink,
Well,
and I shouldn't clean my house.
You can clean the house. Oh, you're right. Well, you can. So anyway, we got 2 wheels now. We got a P53 off. We got an oncogene on. Now a tumor starts to grow. Now, when it reaches the size of about 1/4 of an inch, the outside cells are in this fluid. They're getting oxygen and everything, but the inside ones need a blood supply. There's no blood supply, and if there was never a blood supply, it would stop. We could have thousands of these all over our body. We probably do.
Okay,
But it'll be all right. So a third mutation happens, which teaches a cell that shouldn't to grow a blood system. It starts to build veins and arteries to find blood. And it brings the blood to the cell. Now it's got nourishment, gets rid of waste. Okay, it's called angiogenesis. I don't know if I could pronounce it right. So an obvious cancer thing they tried was a medicine that would stop cells from doing that. Trouble is, it stops normal cells from doing it too. But it does have some help. Folkman was the guy who developed that. He's brilliant.
So that's 4. Are there 2 more?
That's 4. There's 2 more. Okay, so now you got a growing cell. Now, this is beautiful. At the end of every chromosome, there are things called telomeres. They're like shoelace tips. Every time a cell reproduces, it gets shorter and shorter and shorter. And eventually, it can't grow anymore because there's no more telomeres, and the chromosomes blend together, and you have a mush.
This is like the plastic at the end of
a shoelace.
Okay.
Well, the fifth mutation is it learns how to put the shoelace back, the tip. It creates an enzyme, the cancer, called telomerase, which grows it back again. In fact, that would give you eternal life. Cancer cells never die. You've heard about these cells, this woman's cells that were taken from this black woman, and for 70 years they're still growing in test tubes.
Well, they may not die, but we have about 4 more minutes. So.
All right, so the last one. So now I have a cell that won't die, has a bloodstream. Now it gets into the bloodstream. Now remember, if you're a prostate cell or a lung cell, you don't like living in blood. So only maybe 0.0001% of the cells that get in it will wind up at the liver or wherever they're going to, okay? So, but they do it. I mean, they're constantly doing it. So they learn how to survive in that very different pH, temperature environment. And once they're in the blood, they go to where they like to go, like the liver. That's why the liver cancer is rare, but most cancers wind up in the liver, or the bone marrow, bone cancer. Terrible disease. And so the best thing, so now we're developing drugs. Now that we understand this whole sequence of signaling, we're trying to break the signals. You change it. And that's what these new drugs like Gleevec do. They stop it at maybe the second or third thing. So you may wind up with a tumor, but it doesn't grow, it can't get blood, or it can't survive the bloodstream, or it can't replace the telomeres. This is a very simplistic. I'm not a cancer specialist.
Well, you're about here on my
no,
simplistic...
but the point is, it's better to not get cancer than to try to cure it.
Right, and
so
all
stay
those
away from pollution.
All those environmental reasons.
And eat right, and stay thin. Because fat doesn't give you cancer, but fat cells produce testosterone and estrogen, which make the cells grow faster.
Oh, so boy, and here they're selling all these estrogen drugs and testosterone.
Very dangerous. Women who have breast cancer that is estrogen sensitive should never be on estrogen. And when they used to replace them for menopause, it led to, it doesn't cause cancer, but it makes the cell grow faster, which means more mutations, which means it can lead to cancer.
Okay, now there's a lot in the news lately about getting your genome individually. And if you can do that, does that technology, if you know it currently, allow you to know if you have this P53?
It can tell you if you have certain susceptibility. As I said, there's about 500 genes involved, not just in cancer, and it's combinations thereof. The problem with this all is, yes, you could get your genome done for a couple of $100. It used to be $2,000 just 5 years ago. Trouble is, what do you do next? There's no drug for you. See, that's the big problem with cancer. That's why you don't want to get it. Everybody would have a unique drug for them that switched on and off those 500 combinations. How could a drug company do that? I mean, you can think about a doctor's office of the future where you'd sit down, get the genome read, and it would make the drug right there and give it to you.
So we're not at Star Trek quite yet, where they just,
no.
Doc puts that over you.
But knowing how it works kind of tells you what you shouldn't do,
right,
and what you shouldn't eat, and what you shouldn't drink.
Well, I think that's a better way to go, because I don't think my insurance is going to pay for the one drug that works only on me or my family. So
you know, right now they're charging like $3,000 a day for Gleevec, which works. It's one drug for leukemia, but it saves your life.
Now, you're doing some work yourself on this, or
well, I
helping
have
out?
friends. I got my doctorate at New York University, and I have friends that teach in medicine up at Yale. So I don't do anything. I just listen and try to understand it, because I want to make sure my kids don't get cancer and my grandkids. And since it runs in the family, I'm trying to educate them on what they should and shouldn't do. There's so much beyond our control. But it's getting there. We've made more progress in 5 years than everything from 65 up to then.
You are such an interesting person. Not only the things you've done, but just the interest that you take, and then you go forth with it. It's not just absorbing.
I love science and nature.
Go. And then you're trying to do something with it.
To me, it's the only honest profession. You can't cheat Mother Nature.
There you go. So avoid environmental problems, and when you're in retirement, don't just vegetate.
Worst thing in the world, and you got to have social network, like women do.
Well, you're my social network for today. I want to thank you again.
Thank you so much. I appreciate it.
Absolute delight. Thank you so much, Marty.
Bye.
Really appreciate it. Bye.